Good afternoon, everybody. This is a recording of our lecture for osmoregulation and excretion. This is the first part of this lecture. So let me start by sharing my screen, my presentation with you. All right, so we will start talking about osmoregulation and excretion. And this is actually a process or a mechanism of living organism that actually contributes to the conservation of a constant and controlled internal state or environment within the body called homeostasis. So as the name implies, homeo means equal, and stasis means constant or in equilibrium or control. So we can define homeostasis as the permanent balancing and rebalancing of different physiological processes that actually will contribute to keep or maintain the stability of the internal environment, and also they are able to restore this normal state when disturbed. So there are a few concepts I would love to rescue from this definition, which I think is a good definition of homeostasis. First is the concept of ceaseless, meaning permanent. So this is a dynamic process that it takes place constantly, permanently in the body, trying always to keep this internal environment constant. So for an optimum and efficient cell activity. And the other one is that this homeostasis or these mechanisms actually help to restore this internal environment constant state in case it is altered by any either internal or external factor. So the potential changes in this internal environment can actually have or can come from two different sources. The first one is that, first of all, the cellular activity or metabolism needs a constant supply of oxygen, nutrients, and salt from the surrounding media. So while at the same time, cells actually need to get rid of all the waste materials, they have to excrete those waste materials, and they need to remove them in order to keep on working efficiently, okay? And this is, if you think about it, there are some, of course, metabolites, and there are some byproducts of the cell activity that in some circumstances, they are even toxic, and at some concentrations, high concentration, they are even toxic for the organism. So the classical example in this case, and we're going to refer and talk about this a lot, is ammonia. Ammonia at high concentration is actually potentially toxic to the organism. So in some cases, it has to chemically be converted to urea, which is much less toxic, and in some species to uric acid, which is even less toxic than urea, and be excreted outside, taken outside of the cellular environment. So the internal environment or compartment is not actually disturbed, so can stay constantly working and in control. And the second potential source of changes in this internal environment are those that come from the exterior of the cell, and normally and outside the body that actually can produce changes or affect the internal environment. And then the different mechanisms that actually stabilize and contribute to the return of homeostasis will start working. First of all, it is we're going to revisit one concept with this osmosis, which is actually an osmotic gradient, which actually is, we see that normally through a semi-permeable membrane, we will have the passage of solutes, and normally they will move from an area of high concentration towards an area of lower concentration, which can actually be represented by the outside and the inside of the cells, okay, through the cellular membrane. Some of them actually do it by simple diffusion, meaning that there is no energetic cost in the transport, and they just following the concentration gradient, meaning going from the solid will move from the more concentrated side of the membrane towards the side of a membrane where we have a lower concentration. But in some cases, some substances actually have to move uphill, meaning that they need to go from a lower concentrated side towards the higher or the more concentrated side. Okay, and in this case, we are talking about what we call an active transport system. And by active, we are implying that there is an energy cost. And of course, it will be ATP will be involved. Remember, I always refer as ATP as the energetic currency of the organism. So by the use of ATP, we will actually pumping out towards the more concentrated side this solute we are considering. And also we can talk about moving the solute uphill in relation to the more concentrated gradient, and downhill in the case when the solute is moving from the more concentrated to the lower or lower concentration side of the membrane. And one concept that is important, and we will also visit in our next slide, is osmosis. And by osmosis, we actually refer to the movement of water across a semi-permeable membrane, which can actually represent the outside and the inside of for example, the cell membrane could be an example, okay? And in this case, we will see that water will tend to move from a low concentrated, talking about the concentration of solute, referring to the concentration of solute, from a lower concentrated side towards the more concentrated side. So water in general tries to move, we can say that move towards the side of a semi-permeable membrane where more solutes are. So water always tries to dilute the solute, okay? So when, of course, and water will move, aiming to find an equal or achieve an equal concentration of the solute on both sides of the membrane. And when we reach that, we refer as a dynamic equilibrium, that we have reached a dynamic equilibrium for the solute concentration in both sides of the semi-permeable membrane. So what will be the case on, for example, on internal media or the living and non-living organism, or taking as an example cells, in this case, the red blood cells. If we actually place the erythrocytes, red blood cells, in a hypotonic solution, meaning a solution that has lower solute concentration than the inside of a cell, so then water will start getting into the cell, because again, the red blood cells have more solute, they are more concentrated than the media. So water, remember, always move from the lower concentration side to the higher concentration side, trying to dilute it, trying to reach this dynamic equilibrium. So water will get into the cells, and it will reach a point where the actual red blood cells will burst. When we actually place those red blood cells in an isotonic solution, an isotonic means iso means, or iso means equal, meaning that they have the same same concentration of solute, then that means there will be not net movement of water on to any sides, and a net movement, but of course there will be a movement of water until this equilibrium, but both solute concentrations are the same on both sides of the membrane. So there will be no actual net movement of water. There will be no way trying to dilute anything, because they both have the same concentration. And the third case will be when we actually place the red blood cell in a hypertonic solution, meaning that the solution is more concentrated than the cells. So then, of course, water again will move from the lower concentrated side to the more concentrated side, trying to dilute and achieve that dynamic equilibrium. In this case, the cell will start losing water, and of course they will start shrinking and losing the shape. But so the main and the main question we'll have about osmoregulation is, the first one is, why do osmosis and diffusion actually present challenge for animals that live in water? And well, if, for example, we imagine the living organism or the animal like a balloon with a low concentration of dissolved particles, okay? So meaning that the organism has internally a lower concentration of solutes than the media where we're gonna place it, okay? So as we put this balloon, and we imagine that, of course, the balloon is made of a semi-permeable membrane, as using the same examples we gave before. So water will again be moving and start moving from the lower concentration side to the more concentrated side. Again, trying to dilute that side which has more solute. And in this case, the balloon will start representing the animal, will start losing water. And of course, they will start shrinking and become dehydrated. So if we imagine that this is the case of an animal, the red balloon will be an animal, and we are placing this animal, for example, in seawater, meaning that normally seawater has a much higher concentration of solutes than the organisms internally. So of course, this organism will start losing water because water will start moving through the cell membranes, trying to dilute this environment that is more concentrated. And the question is all water the same? Does it make a difference whether the animal is living on a river or is living on an ocean? Of course it's not. Normally in the case of the oceans, we're talking about a media where we are having a hypertonic solution, meaning that it has more concentrated of salts than the individual. And in the case of freshwater animals, they are actually living on a media that is hypotonic, meaning that the media that is surrounding the individual has a less concentration of salts than their body. And so if we think about the other question is, why actually does osmosis and diffusion present challenges for the animals and to live in water? Well, if we move normally from again a freshwater situation to an ocean or to a saltwater situation, we see that the concentration of salts can actually go from 0.05 parts per ton towards a much higher concentration, as high as 35 parts per ton. And this is actually the normal salinity we will find in ocean water. I included this map because it's actually very interesting, and it's referring to the concentration, how the concentration of salt water or ocean or water in the ocean actually changes depending on the geographical area we are considering. And of course, this is going to affect the animals as well, depending on which ecosystem the animal is living. So animals will actually have to deal with that, will have to adapt to the environment where they are actually residing, they are actually living. So the second question is, then how do aquatic animals actually confront the problem of water and salt balance? How do animals actually solve this great challenge that implies the existence of different concentration of or the existence of water with different concentration of salts? So the first type of animals we refer as called the osmoconformers. And in general, marine vertebrates have very limited ability to withstand osmotic changes. This is because they are normally live in ocean waters that do not go through significant changes in salt concentration. For this reason, these animals actually, these osmoconformer animals actually are restricted to live in a very narrow salinity range. And for this reason, they are also referred as stenohalines. And stenos from Greek means narrow, and hals means salt. So meaning that these animals actually live within a very small window, we can say, of variation for solute concentration or salt concentration in water. And this is the case of the sea cucumber, the animal in the picture, that actually passively conforms or accept the external environment. And for this reason, when the internal and external environment present the same salt concentration, we normally refer to this animal as osmoconformers. And again, they do not have the capacity to adjust themselves to big or bigger variation in salt concentration in the water or the environment where they live. And also for this reason, and they actually didn't, by evolution, didn't need to actually develop or acquire this capacity of osmotic regulation, because they actually live on areas of the ocean where the salinity is very stable and doesn't change. So in this case of the sea cucumber, for example, if we actually put them in fresh water, meaning in a water environment, in an environment with a very low salt concentration, then of course, again, applying the principle we just saw with the osmotic the osmosis and the passage of water through a semi-permeable membrane, then water will start getting into the animal, because again, the animal will be actually more constant. This is the freshwater situation, meaning that the solid concentration in the animal is going to be much higher than the fresh water that is surrounding the animal. And then, of course, it will reach a point where this excess of water pumping into the body cannot be excreted, and of course, the tissues will be damaged in such an extent that the animal will die. If we put them, of course, in their usual seawater or salt concentration of seawater where they normally reside, with a concentration, as we saw, 35 parts per ton, which is normally the normal salinity of water in the ocean, then the animal will suffer either nor loss nor gain of water, and that will be, of course, fine. This is another very graphic image that actually shows how the different salinity ranges are represented by the different colors. So we start from, on the left, we have the fresh water, and on the three upper diagrams, A, B, and C, and we see how the different tides and water movement actually produces different fringes or areas of water with different concentration of salt, with different salinity. And of course, that will represent a challenge for an osmoconformer. An osmoconformer, of course, will not be able to survive in this such a highly variable environment in terms of the solid concentration in water. So animals that actually reside, animals that actually live in this environment need some osmotic regulation mechanism to help them survive. So we have here again an actual estuary environment. You know, an estuary is that region where fresh water and seawater actually mix up. It will be the opening of rivers in the oceans, and in those areas normally are very rich in food for example, crustaceans, and of course that will be a great challenge for these animals to survive in this environment, because we can see that how salinity of water will change according to the tide. If we have a low, a high, or a low tide, we will have more or less, we can say penetration of fresh water into the brackish waters or into the ocean, and the other way around, depending on how high or how low this tide is, how much saline water will get into these rivers, into these estuaries. So, so that means that the animals will need to adjust this osmoregulation and through different mechanisms. One, of course, it can be through specialized organs, as we're going to see next, but the other one will be to move. That's why it says one way is by changing their behavior. So that means migrating from the different areas of the estuary, trying to match their ideal saline content of the environment where they actually place or live. So actually they will move among the different environments, adapting to that. That's one solution. So here we're gonna go through an example, which is a case of a shore crab that are normally living in brackish water. So for example, the estuary situation could be a good example. They actually live in those areas where salinity of water will change. And of course, they've been adapted through evolution to live in those areas because those areas offer them more food. So of course, they went through evolution, acquiring different mechanisms to survive. And the other case will be a marine crab that actually lives all his life in very salty water, and is the case of a spider crab. But we will see how both species actually, how much ability they have to osmoregulate in when they are facing different salinity content in the water. So when the internal and the external environment match, as we saw, like the case of a sea cucumber, we call them osmoconforming, or this called osmoconformity. But when the external and the internal environment change, then the animal needs to osmoregulate, need to actually regulate that osmosis in order to keep that homeostasis we talked at the beginning. So if we have in this example, okay, in this first slide, so the first question we ask ourselves is, at 400 millimoles of water, concentration of salt in water in the environment, which of the two crabs are actually osmoregulating? Well, the shore crab, in the case of a shore crab, is what we call a hyperosmotic regulator. That means that they actually have to maintain their body fluids more concentrated than the surrounding water. So for this reason, what they do is they will use their antennal glands, which are similar, or they actually perform a function or a role similar to kidneys. So to do actually that, to actually being osmoregulating, they need to eliminate water that is trying to get into the crab, because again, the crab is going to be more, the shore crab will present a higher concentration of solute than the water. So they actually need to eliminate that water that comes in. And also at the same time, they need to keep their internal concentration of solutes, of salts. And they do that by some specialized cells that are secreting cells that are located in the gills of the crab. And they actually, what they do is they remove the few ions, we can say, that are disposable, that are already in the water that are surrounding them. And then they're going to transport those solutes into their own blood by an active transport. That means by using energy. So in this case, of course, the crab that is osmoregulating is the shore crab. And at 600 millimoles of water in the environment, which of the crabs is going to be osmoregulating? Well, in this case, none of them, because they are both being able to be on a situation as osmoconformers. So the shore crab at this salinity of 600 millimoles of salt, they do not actually need to activate this mechanism of osmoregulation, and they're fine. And of course, the spider crab is fine because he's within the range of salinity they can actually survive. But of course, if salinity went too low, both crabs, of course, will die if we are taking them outside the range where they can actually, the shore crab can actually osmoregulate. And of course, the spider crab is very limited. They have to move between the 400 and the 600 limited. Anything above or below, they will die. So in the case of a shore crab, okay, remember the shore crab was the crab that live in brackish water, in estuaries where there are big changes in the salinity of the water. What are the main challenges that has to face in relation to first, the water or osmosis? Well, again, water will tend to flow into the body of the crab, and it will do it through the gills. And this is because the crab body fluids are more concentrated than solutes outside that correspond to a brackish diluted seawater. What will be the solution when, as we mentioned before, the existence or the presence of the antenna glands, which actually have a role like kidneys. So they will start excreting or pumping the water out. And in this case, they will excrete the water through a very diluted urine. And what about the body cells? So we solve the osmotic problem, through the production of more diluted urine. In the case of the salts, well, as the animal is saltier than the environment, then the solution will actually be so these salt secreting cells that are actually specialized cells that exist in the gills, that through using an active transport against the gradient, remember we mentioned the uphill movement of solute, they will actually secrete the cells in the gills. So that as doing that, they will actually keep the salts constant in their body. And for this reason, we refer to the shore crab as being uri-haline. Urines means broad, and hals means salt, mean that he's able to cope with different or a broad range of salinity. And the spider crab, we will actually call them stenohylines. Steno means narrow, and whole salts, meaning that spider crabs are only able to live within a very narrow range of salinity in the ocean. And if we look at the blue curve that corresponds to the osmoregulation the shore crab is able to perform, we can see that they can even survive in very low saline water as low as 200 in comparison by the spider crab, that he can only survive down to 400. So at 200 concentration, millimoles of salt, concentration of salt, the shore crab will eventually survive, while the spider crab will be dead by then. It's impossible he can stand at 200 concentration in water. Now, similar situations we will see in the case of a freshwater bony fish, which are the challenges of these fish which actually live in rivers. Well, they actually, the fish, of course, gonna be saltier than the environment, meaning that in their body, they have a higher concentration of solutes than the water surrounding them. So because of that, they will need to recover salts that are actually lost by diffusion from their body to the water that has a lower concentration of salt. And at the same time, they actually need to excrete the excess of water that is trying to, by osmosis, get into their body in an attempt, again, to dilute the higher concentration inside the body of a fish. So what are the solutions? Well, from a physiological point of view, will be the active uptake of salts in the gills by some cells called chloride cells, and also by the kidney. But how we're going to do that? Well, also the gonna produce a more diluted urine. So they will actually excrete a lot of water in their urine. And from a behavioral point of view, well, these animals will very seldom, very rarely drink water, okay? So that's also a way of preventing the excess of water coming getting into their body. And we call these animals hyperosmotic regulators. That means that they are actually keep their body fluids, they are able to keep their body fluids more concentrated than the surrounding water. Now, in the case of a saltwater bonyfish, which is going to be the opposite situation, the challenges these animals are facing is that these fish are going to be less salty than the environment, so there will be more solutes outside their body than inside their body. For this reason, of course, water will tend to leave the fish, trying in an attempt to dilute the environment. So these animals, the main challenges they will be facing is to retain water and, of course, get rid of the excess of water that will be getting through their gills. So what are the solutions through evolution these animals have achieved? Well, from a physiological point of view, they will perform an active excretion of salts at the gills by chloride cells, and the kidney, they will be able to concentrate the urine, and they will produce a very salty urine, but with a very low volume of water. So by doing that, they are actually saving or retaining a lot of water, so they actually do not become dehydrated. And from a behavioral point of view, well, of course, as the opposite situation of a freshwater bonyfish, these fish will actually drink water as compensatory mechanism from their behavior, from a behaviorist point of view. And these animals, we call them hypoosmotic regulators, meaning that they are able to keep their body fluids at a much lower concentrations than the surrounding water. Well, similar situation than the freshwater bonyfish, we have the frogs or amphibians, and we know they live in water, so they need to actually compensate for the loss of salt, who will try to diffuse from the body into the surrounding water, and also they need to compensate for the excess absorption of water, because like the freshwater bonyfish, their body fluids are going to be much more concentrated than the surrounding water. So they will need to actually absorb salts and excrete water. So in the case of a frog, and it has been shown that in the skin, they are actually perform a very active transport of sodium chloride, and the kidneys of the amphibians are actually able to produce a very diluted urine by the reabsorption of sodium chloride, but also they excrete urea and ammonia. And urea and ammonia, as we know, they actually drain or they draw a lot of water when they are being excreted. And that's also a way of actually eliminate the excess of water that is by osmosis getting into their body. And because of this very active transport of sodium chloride they have on their skin, that allows them to absorb salt. The skin of a frog probably is one of the most studied models for the sodium and chlorine transport through tegument. And also the bladder will be able to reabsorb the remaining sodium chloride and will be returned to blood. So that's another mechanisms they have to actually absorb or conserve salts. So let's have a look at other animals. In this case, we have the sharks and the coelacanths, and they actually are able to bring the internal solute concentration of blood and body water up to or slightly above that of seawater. So actually what it means is they are able to reach equal osmotic concentration, osmotic pressures inside and outside. So they actually are able to keep a water balance. So by doing that, the osmotic difference is eliminated between the blood and the surrounding seawater. So water balance actually doesn't represent a problem for these species as they will be in an osmotic equilibrium with their environment. So how they do that? How they achieve this? Well, they do it by the retention of solutes, in this case urea, and trimethylamine oxide, which is a compound they are able to synthesize. So these are waste product. Urea is a waste product. And they are actually retained, and they actually use as osmolyte. So they use these products of excretion to regulate their internal osmotic pressure, and by doing that, they reach an equilibrium with the surrounding water. And other species, for example, marine reptiles and birds, how they actually survive? Well, they are hypoosmotic regulators, meaning hypo means low, and osmotic, of course, refers to osmosis, osmotic pressure. They actually are able to maintain their body fluids at a much lower concentration of solutes than the environment, than the seawater environment. And this, in the case of the marine birds, they actually, and reptiles, they actually present the salt glands, which are glands that actually are able to excrete the excess of salt, and they are actually modified lacrimal glands, and they are able to excrete 2 times a saltier fluid than the seawater, or 6 times saltier or more concentrated than blood plasma. And in some cases, it actually achieves kind of, almost jelly-like consistency, and that is actually a very effective mechanism. So they are able to get rid of the excess salt they actually have to face in their environment. In the case of marine mammals, well, the kidney of these animals is actually a very highly specialized organ, and by using the kidney is able to carefully regulate the salt and water balance. And of course, in this case, they do not drink water, so most of the fresh water they need for the sustainment of life, they do it from, or they take it from the food and from the metabolic water. Remember that for example, the metabolism of lipids and complete oxidation of more nutrients, we have the production, for example, of water, and that's one of the origins of the metabolic water. And then we've gone through all these aquatic organisms. Either we saw the case of the freshwater crab and the marine crab. We saw the situation of the freshwater and saltwater bony fish. But what are the challenges that actually those animals that are terrestrial, that live on land, have to face in terms of osmoregulation and excretion? For example, limited evaporation, that could be one. So there are 3 main areas we will see that evaporation is going to be controlled or limited in a way to save water from their organism. From a morphological point of view, they present normally a thickening of the epidermis, and the keratinized structures like hair, scales, and feathers help to prevent the loss of water. And they also, some of them present actually lipids in their epidermis, or they present actually mucus secretions that actually prevents the excessive loss of water. From a behavioral point of view, well, they will actually alter or change their behavior, their activity, like, for example, trying to be more active during the night when the temperatures are lower. So that's also a way of saving water that they might be losing through sweating or perspiration. And also time of the year, sometimes animals are more active or less active during the hot or the colder months of the year. So in this case, let's say in the case they want to limit evaporation, of course, they will select the cool and more moist or high moisture habitats to live. And from a physiological point of view, well, they will actually present or are able to sustain a low metabolic rate and breathing rate. And we're going to use as an example the kangaroo rat, that is a rodent that actually lives under very dry and hot conditions in the desert. So we see that from a behavioral point of view, as a way of saving water, because of course they live in an environment where water is actually very scarce. For example, the animal will remain inactive during the hottest time of the day, normally living in burrows, in caves. They will actually, that will allow them to waste less water through the respiratory moisture that is normally condensed in the nasal passages, trying to cool down that air that is coming into their nostrils. And also they will get the metabolic water from their feed. They will try to save as much water as possible in the feces. For this reason, they will actually produce very dry feces pellets. And of course, they have this ability to concentrate the urine by using the countercurrent mechanism we will see in our next slides in the loop of Henle. In this case, these animals, we have a really long loops of Henle that actually allows the urine or the fluid that is being traveling through the ducts in the kidney to actually go through a much wider gradient of solutes, and that's a mechanism the kidney has to concentrate urine. But we will see that with more detail. So in this table, which is very interesting, we are actually comparing the situation of the human and the kangaroo rat, and we will see that if we add gains and losses, of course, they both add to 100%. And so if we make gains minus losses, will give us a 0%. So that means that these animals, the situation where both are in equilibrium in their water homeostasis. And in the case of a human, 48% of gains of water are achieved by drinking water, from drinking water, while the kangaroo rats, 0%, because of course they live in an environment where they do not have access to drinking water, to fluid water. So 40% of the water the human acquires comes from free water in the food. In the case of a kangaroo, it's 10%. Normally they eat very dry feeds, food like nuts, for example, of this kind of food that actually doesn't contain much water. But here comes the first big difference. It is the case of that human is able to obtain only 12% of their gains of water, or their water intake of water, by the production of water through the metabolism or metabolic routes or pathways. In the case of the kangaroo rat, 90% of the gain of water or the gain of water they get in their body is through metabolic water. And when we look at the losses, we see that the human he will lose about 60% of water through the urine, where in the kangaroo rat, it will only represent 25%. In the case of evaporation, lungs and skin, 34% will happen in the human, while 70% will be the losses in the kangaroo rat. And this is, of course, because they actually have to survive in this very dry and hot environment, and they need to dissipate a lot of heat. And if we see the feces, the loss of water is not very different. So we see 70% of the water loss of a kangaroo rat will be through evaporation of lungs and skin, 25% only urine, so they are able to produce a highly concentrated urine thanks to these extra long loops of Henle, and 90% of the water they gain or they get comes from their metabolism. And with this slide, I finish this first section of osmoregulation and excretion. Thank you very much for your attention.